Today, we are lucky that we have two people from MSU here to talk to us. Benjamin is going to, from our cropping system agronomy, he's going to talk about some of the research that he was compiled and put together on some work that our field crops team, led by Dr. Eric Anderson, did in Michigan throughout this past year.
All right, thank you, everyone for joining. I apologize for being late. I was looking for the Zoom link, but I wasn't finding it. Yeah, so I'm glad to be here today to share the results from this work that I've worked on for the past, I would say, 2 years. And we were for this so I'll start this way. So my PhD research is looking at how we can improve sustainability of agroecosystems in the Midwest and to a larger extent the northern production systems. And so what I've been focused on is how to identify some management strategies to first of all improve soil health, and then that could lead potentially to some yield benefit and also to some profitability. And soil health is big. Soil health is huge. We have different components of soil health. We have the physical component, we have the chemical component, and we also have the biological component. And most often than not, a lot of research has been, most of the previous research on soil health has mostly been focused on the chemical and the physical aspect. And we don't have so many studies on the biological component. And one part of the biology or the soil biology, which is super important, is the soil microbial community structure. So right from here, I'm showing this diagram indicating some soil microbes in the soil. We have several microbes in the soil. If you take a gram of soil in your hand, there are millions and millions of microbes within there. And these microbes perform super important functions. If you look at the figure over here, it's illustrating some of the key functions that soil microbes perform. And one of the most important functions is what we call the nutrient cycling. So most of the nutrients in the soils are in the organic form. And so these organic nutrients have to be mineralized for them to become inorganic for plants to absorb. And most of these dynamics are done by some of these soil microbes. Apart from nutrient cycling, soil microbes are also important in plant growth regulations and stress tolerance. They also help in pest and disease control. They also help in degradation of pollutants. And so the microbial component is super, super important. And there are several ways to quantify soil microbes. You can measure, you can estimate some properties that illustrate microbial function, and you can also physically quantify how much you have in your soil. And so a lot have been done on identifying the measurement that illustrate microbial activity. And so it's common to find research on measuring some indicators such as POXI, which is like permanganate oxidizable carbon. You might also have read some or seen papers talking about soil proteins and all those ones. So these are all indicators that illustrate microbial functions, but it's also important to quantify how much we have in the soil. And that has been the focus of this research over the years. And so for the objectives, we wanted to understand the main drivers of soil microbial community across Michigan farms. Because the soil microbes are affected by multiple things. Some of them have to do with the soil properties, such as the texture of the soil, the structure of the soil, porosity of the soil. And the other dominant thing that also affects soil microbial community is management. We have diverse management practices across farms. Some growers are under no-till, some are under conventional till, some are under strip till. We have growers who plant cover crops after harvest. There are growers who don't plant cover crops. There are growers who rotate corn with several other crops like soybean, wheat, and all. And there are growers who do continuous corn. There are fields under perennial alfalfa that fall under several other things. And so all this management affect your soil health and most importantly, your soil biology. So if you're under continuous corn row production, your soil biology will differ from if you're under corn soybean rotations. But we don't have a lot of studies examining these relationships, especially using on-farm data set. And that's one of the unique things about this research that we did over the years. So we are using on-farm data set to report how different soil biology in terms of microbial numbers are across Michigan. So our main objective was to understand what are the drivers by looking at some specific things that we measured. So our first objective was to see if we have differences in microbes based on the soil texture. And so if a grower is under, if the texture of a soil has more clay, are you having like more microbes as compared to a soil having more sand, or compared to a soil having more silt? And so that was one of the main specific objectives that we wanted to test. In addition, like I mentioned before, all these things are affected by management, and the two key management that we wanted to evaluate are your crop diversity. When I mention crop diversity, I'm talking about how many crop species do you plant in a year. So for example, if you grow corn and then after corn, you put a cover crops, you have more diversity as compared to if you grow corn and don't put anything else after corn. And so if you have corn, soybean, and also a cover crop if you have double crops and also a cover crop within the same year, you have higher diversity as compared to someone with just corn and soybean without any cover crops. And so we wanted to understand if you're able to fit more crops within a rotation, are you getting benefit as compared to having single crops or just two crops within the season? And finally, one of the main drivers of, I would say, soil organic matter, soil health is tillage. So the idea with tillage, or the mechanism with tillage, is that if you till your soil, you break these micropores and you break your soil pores. So first of all, you destroy the soil structure. And one thing about soil structure is that we have these micro and macropores. So the function of the micropores is that they are able to occlude or protect your soil carbon from rapid decomposition by microbes. And all these things helps in carbon accumulation, which is helping like some of these things that you have been talking about, this climate regulation and all. And so if you continuously till your soil, it has its own benefit, but then for the most part, you are harming your soil structure, you are harming your carbon accumulation, and it could also be harming your microbial dynamics. And so we wanted to understand how tillage affects our microbial numbers. So this research involved like soil sampling across multiple locations. So the figure over here shows all the counties, and within the county, there are specific fields that were sampled. But we were able to take soil samples from 29 counties, and most of the work was done by Eric, Christine, and some of the other extension educators. I wasn't part of the soil sampling because I wasn't in Michigan living at the time of the sampling. And so the sampling began in fall of 2019, and the sample was taken on fall 2019. Samples were taken on spring 2020, and fall 2020, and then the last sample were taken on spring 2021. And this large number of sampling is to help us to have more confidence in the data, because the more you have, the larger your sample size, the more the rigorous your analysis, and the more confident you are in your data set. And like I mentioned, we took samples, or the samples were taken from multiple fields. So on the average, we're between about 65 to 86 fields across all the sampling times that we did. And so the main focus, like I mentioned before, was to understand the microbial numbers, how much microbes we have in the soils. And there are several ways that we can quantify how much microbes we have. And one of the cheapest way, or the most affordable way and easiest and rapid way to do this is what we call the phospholipid fatty acid analysis. My next slide will talk about this in a little bit more detail, so I'll just hold on that for here. And so after taking all these soil samples and taking them to the lab for the analysis, I came in Michigan, and then we developed this field management history. So the idea is that I wanted to understand how growers have been managing their fields before the time that we took the sample until the time that we took the sample. Because the idea is that how you manage your field is going to impact your soil microbial populations. And so we developed this field management form. We sent it out to the growers, and we asked for their rotation, how much, like what is their rotation like, Are they under continuous corn? Do they rotate corn with soybean? Do they even put wheat as part of their rotation? And we took this data from 2016 to 2019. And then so growers sent us those forms. So growers are asked to indicate their crop rotation. They were asked to indicate whether they are under conventional till or they are under no-till, and also how many tillage passes they do within a growing season. And then we also asked for their field coordinate. I think, Eric and the other extension educators who took the soil samples always took the field coordinate of where they took the soil samples from each field. And so with those field coordinate, I was able to pull the soil texture data from the Soil Web Survey. And the idea is simple, or the mechanism is simple. So you log on to the Soil Web Survey and you put in those coordinates, and you're able to draw your area of interest, which is closer. So this is the pin that those who sample the soil will draw. So with this pin, I'm able to draw this area of interest here, and I'm able to extract the soil texture data. So I get a percent clay, I get a percent sand, and I get a percent silt. And with that, we're able to develop these soil texture classes as to your fine or your medium or your coarse texture based on the percent clay that you have in the samples. So this was a post, and the texture was not determined as part of the package in the analysis, but then this was extracted from the Soil Web Survey. So like I mentioned before, our main goal was to quantify how much microbes we have in the various farms that we sampled. And the approach that we use is the phospholipid fatty acid analysis. So I wouldn't go into all the details, but the process is quite simple. So you pick soil samples. So Eric and Christine and all the other educators pick the soil samples. They store them under in coolers just to reduce microbial activities and then these samples were transported to the lab. And the analysis were done in the soil health lab in Cornell. No, I think in Missouri. Yep, I think in Missouri. Yeah, and so for the sample, when the samples are there, it goes to these chemical preparations. They extract the phospholipid fatty acid. And just to give a little bit details, so each soil microbe have what we call the phospholipid boundary layer or membrane. And within those phospholipid membrane, they have these specific fatty acids. And each microbe, for example, bacteria has its own characterization in terms of the fatty acid. So fungi also have its own characterization in terms of the fatty acid present. And so we use what we call the biomarkers. So these biomarkers will identify the specific fatty acid within the bacteria and specific fatty acid within fungi, within AM, on all these broad groups. And these are broad groups. These are not species specific. These are broad groups of bacteria, like broad groups of microbes. And so once the bioassays are able to identify these specific microbes, we take, after all the extraction and all the identifications, we have what we call the gas chromatography. It's a big machine that you put your samples in and you're able to identify those biomarkers to match them to the fatty acids that are specific to these groups. And then you spit out the numbers and you're able to quantify how much of these broad groups of bacteria, how much of Lactomycetes, how much of each group that you have in the soil. And so the difference between the PLF analysis and other DNA sequencing analysis is that for PLFA, we have this broad group. These are bigger groups. These are not species specific. Because if you take bacteria, we have several species of bacteria within the soil. But then the analysis will quantify the bigger group of bacteria within the soil. So that was the process. And the output that we were mostly interested in so the total microbial abundance is overall how much microbes do you have in the soil. So this indicator shows the total amount that we have. And then in terms of specific groups of microbes, we have these bacteria, you have the Actinomycetes, you have fungi, we have AM fungi, you have eukaryote. All of these broad groups of microbes perform specific function in the soil. Like I mentioned before, some of them are involved in nutrient cycling, some of them help to break down or decompose organic matter for nutrient release. Some of them help in regulation of cycles in the soil. Some of them help in plant stress tolerance. And so these are super useful microbes and super functional microbes within the soil. And so overall, that was the analysis that we did, and the data came with all these broad groups. And now we analyze the data in our studio. I did all the analysis, and our main, like I mentioned before, the main interest was to understand how texture and how tillage and how crop rotation or crop diversity affects some of these numbers. And so the first result here is showing the effect of soil texture on these microbial groups. And so the x-axis here shows the soil texture. And here I'm not reporting the percent sand or the percent silt or the percent clay over here. So we, based on the clay fraction of the texture, we have these three groups. So if you are quartz, the quartz soils have the least amount of clay. The medium one has I would say moderate amount of clay, and the fine soils have the highest amount of clay. And so the y-axis shows the numbers of each. So the first graph over here shows the total amount of microbe in each soil, and then the other 5 graphs shows the individual amount of each big group of soil microbes. And so if you see for the first graph, the total amount of microbes, we really did not see any significant difference between the three main soil classes. So soils with less amount of clay were having equivalent amount of microbes total amount of microbes compared to soils with high amount of clay. But then the interesting part is when we get to some of the specifics. So we realize that the only microbial group that was significantly impacted by the soil texture are the actinomycetes. And so you see that soil with the highest amount of clay gave us the highest amount of mostly had the highest amount of actinomycetes. But for the other ones, but we also found some trend with higher microbial populations with high clay content. And the idea is that clays have high surface areas, and so they're able to hold more soil nutrient because of these negative charges on the clay, which are able to hold more of these nutrients. And so these nutrients helps in the assimilation by the microbes, and so it's able to influence the microbial numbers because they have high amount of nutrient that the microbes can use even before they are able to degrade the other numbers. And so it's also important to mention that several other studies in different locations have identified that soil texture significantly is able to impact your microbial population. And I feel that, the observation here, given that this is regionally specific data, gives it's kind of positive in the sense that growers understand the soils shouldn't really be much bothered if they are able to improve the other management practices, because this data is showing that your microbial numbers is not significantly impacted if you're under much sandier soil as compared to someone under more clay soils. And so it's important to understand what other mechanism in terms of management is able to impact your soil microbial dynamics. And like I mentioned, soil microbes are super important part of the whole soil health process or the soil health concept. And so if you're able to improve your microbial dynamics or your microbial numbers, you are likely able to improve more of your soil health. And so essentially, growers under much sandier soils compared to those under more clay soils can all improve their soil health to a significant extent. And so for the two other crops and for the two other management practices that we wanted to test, so we had a crop diversity. So for the data that we asked for, we had 3 years of rotations. And so if you see one, so on my x-axis, I have the crop diversity, and on the y-axis, as the previous graph, I have the individual microbes and the total microbial population over here. And so 1 stands for a field which was under continuous corn. So it was corn throughout the 3 years of the data that we collected. 2 stands for a corn, soybean, corn rotation over the 3 years. So there was corn in the first year, corn in the second year, and then corn in the third year. And the 3 stands for corn, soybean, and then wheat kind of rotations. But sometimes for not just corn, soybean, and wheat, we also found that there were other crops also coming into the rotation, which were not just wheat. And so there's more things happening over here than the previous graph. If you see here, where once again, our microbial numbers were not significantly, the overall total was not significantly impacted by a rotation type. And sometimes this makes sense because most of these things, in terms of crop diversity or having crops in the ground, it's all about biomass production. And so if you're able to produce adequate amount of biomass under corn even under continuous corn, and you are not, then that means you have a lot of substrate that the microbes can use, because the microbes depend on some of the substrate. And so if you're able to produce enough amount of biomass, then it's able to offset whether you are under continuous production of the same crop or you are rotating with other crops. But that has to do with the overall. But one thing to also find is that we should also be mindful that there's like a trend of higher microbial numbers with increasing crop rotation. But this is just a trend. It wasn't significant. But another thing that was interesting is that we found significant effects on some specific microbial groups. So if you look at A.M. fungi numbers were super high or were significantly higher under more complex rotations, where you have like at least 3 different crops in your rotations compared to when you have like single, or you have like 2, or you have 1 rotations. And in terms of their specificity, these different microbes in the soil respond differently to the kind of biomass present. And so some microbes will want to have more affinity to work on biomass that has high amount of nitrogen, like the legume ones, like soya beans. And so if you have some of these come into your rotation, then you're able to attract some of these microbes. And that's why it's always meaningful to pay attention to your rotations, because it affects the specific microbial groups and might not even affect your overall, the total amount of microbes within your soil. Like I mentioned, so the total amount of microbes is a function of how much biomass you are producing. That's the starting point. And again, the specific microbial groups depends on what type of crop, because they have affinity to different kinds of biomass. So some microbes prefer some soils, some biomass from legumes. Some do not have, some have less affinity to kind of discriminate against the kind of biomass that is present in your soil. And then we also find a trend of increasing microbial numbers with increasing crop diversity. And bacteria is one of them that had that trend. And bacteria is super, super important in the soil. It's helping carbon mineralization, and this also helping a lot of these nutrient cycling processes in the soil. And so finally, we have the main guy, the bigger one, which was the main driver of the soil microbial numbers in the soil. And so tillage significantly affected the total amount of microbes. It's also affected all the microbial dynamics. So you can see over here, the total microbial numbers overall was significantly lower under the tillage than the no-till. And like I mentioned before, tillage really destroys your soil structure. It breaks down aggregate. It exposes your hidden carbon to microbes for them to work on it. And so that was the main thing that we found. And if you look at the numbers were super, super low. If you look at fungi, if you look at bacteria, if you look at Actinomycetes, their numbers were super, super low under no-till than under conventional till. Sorry, they were significantly lower under conventional till compared to the no-till. So to conclude, one of the main takeaways from this study is that in Michigan, the dominant driver based on our research is tillage. We found tillage to be the dominant driver of soil microbial community dynamics across the farm that we established. And as I must also mention that our study evaluated tillage, and we did not take account the number of tillage passes because the data that we got were not adequate enough to be included in the final analysis. And so were based on whether it was continuous no-till or it had at least some tillage practices happening. But based on our results, we can say tillage is the main dominant driver for soil microbial dynamics across Michigan farms. And then we can also say that we found some effects of tillage and crop diversity, but those had minimal impact. And so to provide some recommendations to growers going forward, we can say, given that tillage is the main driver that we found in this research, we can say, switching to some conservation tillage practices to reduce or minimize how much disturbance you have on your farm or on your field will go a long way to help build your soil microbial dynamics and also build the overall soil health. And some of these minimum disturbance can be reduce the number of tillage frequency, reduce the number of tillage passes, or if you're under conventional till and you want, you can even switch to no-till. And these have shown that there's great potential to build soil biology and overall soil health in these systems. And given that we have some effects from from crop diversity, we are also encouraging growers to maximize biodiversity. You can include cover crops in your rotation if you're able to fit them. And there are so many questions under cover crops as to which species you have to put, when you have to plant, how do you get adequate biomass? These are all questions that research is trying to solve. But then we have shown that if you increase some of the micro have shown that if you have higher biodiversity, and the biodiversity can be achieved even if you have cover crops in your rotation, not just rotating between corn, soybean, and wheat, but you can also have cover crop within, and even have more cover crop mixes. Having higher cover crop mixes also add to your biodiversity within the growing season. And you also have to increase crop diversity. I mean, moving from just continuous corn to corn soybean system if possible, and also have the integration of livestock. And all these have been conceptualized into a framework that we call the soil health principles. So the overall idea of the soil health principle is to maximize soil cover, to minimize disturbance, to maximize continuous living root, and to maximize biodiversity. And these are some great ways that you can achieve some of these principles to improve your soil biology and also improve your overall soil health. So to conclude, and I'll be ready to answer any questions if there are, I would like to thank the funding agency, MDAT, and also thank all the extension educators who did great job taking all the soil samples from all these different locations across Michigan for all these years. It was such a hard work, I believe, for them. And to also give some perspective, this research have been published on the Soil Science Society of Agronomy Journal, and the title over there is Farm Level Variability in Soil Biological Health Indicators in Michigan is Dependent on Management and Soil Property, like I've briefly summarized over here. And so if you want more in-depth explanations, you can go to this website and then find this paper, and you can read to get more explanation to what we have. We also put in Michigan and MSU extension bulletin, and this is the bulletin number. I believe it will be out very soon. And over there, we are summarizing the same research over there and giving some recommendation out over there as well. And if you want more information on soil health and some of the work that we do in my lab at the Cropping System Agronomy Lab, you can visit our website. This is the address for the website, and this is showing what you see when you visit our website. And you can go there and do some further readings and ask questions if you have them, and we will be ready to respond to you. Yeah, so I will end here and take any questions if there are any questions. Yeah, thanks everyone for listening to this talk.
All right, there are a couple of questions for you, Manny, and while he's answering the questions, I'm just going to flip up a quick poll. I'm going to try something new this month and see how this works. But the first question is from Scott. His question is, which of the microbe groups have helped make phosphorus available for crop uptake?
So off the top of my head, I would say AM fungi has been noted to help in phosphorus uptake in the soil. So AM fungi will be my first answer. But then these microbes perform multiple functions. But the one off top of my head will be AM fungi.
So Keith is asking, have you compared any treatments that are perennial grasses and or legumes to annual row crops?
So there are a lot of studies on that one, but our work did not focus on that because we did not really get enough data set to make those comparisons. That's what I would say. So everything was annual system. We didn't have a lot of data on perennials. There was some field report that reported like alfalfa having multiple years of growing, multiple years of alfalfa, but then those data was limited that we could not perform adequate statistical analysis to report on that.
Thank you. Scott's asking, do you think there is a difference in microbes associated with thin fibrous roots and tuberous roots? Have you seen a research addressing this question in your study sources?
So I've not delved much into examining some of these thin fibrous root and tuberous root, but like I mentioned before, so the biomass, which include above ground biomass and below ground biomass, will affect specific microbes. Some microbes I believe some microbes will be, because it's not just about whether it's thin, or it's about the chemical composition, how much you have in them, because we have what we call the CN ratio. So microbes will want biomass that has more nitrogen so that they can use that one to also break down the carbon. And so if we have differences between the carbon to nitrogen ratio between the thin fibrous root and the tuberous root, I believe some microbes will have affinity for these different kinds of substrate, I would say. But I've not really seen, or I've not delved much into any research that really tackles this specific question.
Okay, thank you. Mike would like to know if your plots were tiled, and if so, what was the spacing?
So for that, I think Eric can help, because I personally did not go to any of the fields. It was Eric, Christine, and some other extension educators who did the soil sampling. And so if Eric have any ideas whether some fields were tiled or they were not, Eric could probably answer that.
Yeah, so these weren't plots per se. These were just grower fields, and we did not prescribe anything about the fields, rotation, soil type, tillage type. I don't even know that we have data on whether or not the fields were tiled. We may have that in some of our history, but I don't know it off the top of my head.
Okay.
I will agree. I had 2 that were and a couple that weren't. So it just depends on your field. So Mark is asking, for non-irrigated farmland, are long-term crop yields and total biomass a good passive measure for soil microbe quality and soil health?
Yeah, so that could be a way, but you also have to be mindful of the fact that sometimes what we call the nutrient mining. So you can be having, or you could be seeing some increase in yield over years under non-irrigated conditions without doing anything that is geared towards improving your soil health or your soil biology. And so even though you are seeing increases in yield, that might be that your crops are, or the corn or the soybean is mining the residual nutrient in the soil. And so that might not be sustainable in the long term. So a good way to see whether you are really building soil or doing something good is to take soil samples and test them. And like that cheaper and there are less expensive indicator that you can measure that can give you good assessment of how or the status of your soil. So yeah, I agree. It could be a good passive measure, but then you have to be mindful that you are not mining the soil rather than just or something like that, I would say.
So Dan is asking, would soil genomics been more revealing? Those results are fascinating when comparing landscapes and over time, sorry.
Yeah, so genomics will definitely shows more of the diversity within each microbial group. But those ones are much more expensive and takes more time to really estimate how much, because they'll have to be like DNA extraction and all those things. And the idea is that, so the good thing about PLFA is that you're able to understand what is the biodiversity. So first of all, have to estimate, are the microbes even present in the soil? And if you are there, what functions? So we are answering the first part of the puzzle, which is, are the microbes even there? So the genomics part will give you more of the specific species within the soil or within the microbial group. Yeah, that I believe will be much more, yeah, but that will be the second step of some of these things that we do. But PLF analysis will give the idea whether the microbes are there or the microbes are not there. And I believe that's a super useful information, because you can use that one to inform immediate management practices. Or like that one could have more immediate results than just focusing on some of the specific species.
Okay. So Benjamin, Ben is asking, fungi is slow growing. Is 3 years enough to see a big difference?
Definitely, I would say not enough. I mean, the idea with data synthesis and all this collection is that we can look at a wide range of data that we could have done multiple years. And like we could have done like 5 years. But it also has to it goes back to the growers. How much are they even able to keep? How much are they able to remember to provide? And so we believe, or I think one of the ideas why we limited our study to this 3 years is that so that we can get the one that the growers can easily remember, and then we can work with those ones. But I agree, fungi grow slowly, and so increasing the number of years could have potentially give more results. I agree.
Yeah, and just to clarify, so this is not a longitudinal study. So it was done over 3 years, but each of the sites was not sampled each of the 3 years. So each of the fields was chosen in the given year. It was not sampled each year. So for example, I might sample this particular field that I'm in front of this year, but if I'm working with this grower next year, we're picking a different field. So we're just looking at, it was really just 2 years that the study was done, 2 replications. So we're just looking at these as snapshots in time, not as longitudinal.
Right.
So Dan is asking, did you correlate with pH, EC, and active carbon with responses? In my experience, along with the structure have a big impact on microbial community.
Yeah, so we did some Pearson correlations, and that one is reported in the paper. So we correlated. I did not correlate with pH and EC, but we definitely did some the active carbon, which in this study we measured epoxy permanganate oxidizable carbon. We also correlated that one to soil structure over there. Yeah, we have WSA, which is water stable aggregate, which shows your soil structure index. And so pH wasn't part of the correlation metric that I did, or I think maybe it was there. Yeah, if I remember correctly, I think pH was also part of it, but we didn't have EC for that. So in that paper, if you read, we saw some positive correlations with some of these microbia, especially epoxy, which is like active carbon. It correlated with some of the microbial dynamics that we found. And that is in the paper that we just published.
Nick is asking, is there work being done looking into the role of cover crops and the rotation in these biological communities, specifically looking at the effects of species, planting, date, termination, timing?
Yeah, there's a lot of materials out there, but the approach is mostly like I mentioned. Some of them, so we have for microbial assessment, we have 2 things. Either you measure the microbial numbers, like we did over here, or you measure some activities that reflect microbial functions or microbial presence. So there are a lot of studies that have looked at how epoxy, how soil protein, how soil enzymes, which are indicators of soil biology. And so there are a lot of papers out there indicating how species and timing, biomass accumulation, and all affect these microbial communities. But you have different kinds of papers, and sometimes you can correlate the functions or the activities to some of the numbers.
So Hillary wants to know why we use PLFA rather than microscopy.
So, like I mentioned, we have different ways. So, microscopy, do you mean so Eric, so for my understanding, like I mentioned, PLFA is because one thing about soil health indicators is that it should be, first of all, you need to think about the cost and then how effective those ones are. And so I don't know how much microscopy will cost, but I think PLFA gives reliable data. The data is super reliable because you have actual microbial numbers which are identified based on the phospholipid membrane layer. And so we have this one. I mean, the downside is that we just bigger groups and not specifics. And so I feel like I think the cost side would have also played a role because there was a lot of samples, and you also have to think about how much you are also paying for that. I don't know if that really answers the question.
I think Eric and I can expand a little bit on that. We were asked to do PLFA samples by our Department of Agriculture and Growers. They were curious to see what those actually were, so that's why that's why we went with that testing.
Okay.
And Anne would like to know if there's any impact. What is the impact, if any, of synthetic pesticides and fertilizers on biodynamics within soil versus more organic inputs? So
I think there's this. So we have ways that we can build soil health. I mean, some people consider soil health to be a semi-open or semi-closed system. So if you say it's a semi-closed system, we allow some input. And it's all about the dynamics. Like I mentioned, nitrogen is one of the key things that microbes need. And so if you put some fertilizers, if you put like urea in there, urea will give readily available nitrogen for the plant. for the microbes. So the microbes can use that, and they might not be mining the soil, the residual nitrogen within the soil organic matter framework or the soil organic matter pool. And so, but then those ones are transient, those ones are they are rapid. And so if you continuously put fertilizers without having any organic input, then you really do not gain by increasing the numbers. Because if you put urea, the plants will just take them, the microbes take them, break them down, the plant, the corn or soybean or whatever will absorb them. And then you are not really having any additional input. So you gain more by having organic input, having more biomass, having residues on the soil, having more cover crops in your rotations. You terminate them and still leave the biomass over them. And so organic input versus synthetic fertilizers or some other synthetic chemicals, those synthetic chemicals, I would say their turnover time is super fast. Less than a month, they are gone. But if you have some of these organic inputs, their turnover time can take months to years. And so you really gain there by having those ones there and having something for the microbes over a long term. And so you are able to increase your microbial numbers with these organic inputs.
Thank you, Benjamin, for giving us great information.